All right, thanks, Ami. Thanks to everybody who came. And as Ami pointed out, this is a lecture not sort of geared towards card-carrying neuroscientists, because a lot of it, especially the first part, is kind of textbook knowledge. You can just read any textbook and find some of this information. I present it because I'd like to believe I contributed in important ways to that textbook knowledge. And so it's a talk that I've given over the years for grand rounds in psychiatry, to clinicians, to scientists who are not neuroscientists, to lay people. So that's the level it's going to be at, and the title is here. And I think when most of us, or at least when I think about the brain, or for that matter, nervous systems, one of the most fascinating and important properties of our brains, and for that matter, any nervous system, is its plasticity. And what I mean by that is the ability of an experience, whether that experience is sitting in a classroom listening to a boring lecture, whether that experience is ingestion of a drug, whether that's experience is a stressful event or a car accident. It's the ability of that experience to modify the hardware of the brain, and as a consequence, modify our subsequent thoughts, feelings, and behavior. While I think there's certainly a genetic component to certain aspects of our personality and who we are, for the most part, we are who we are, we do what we do, we feel what we feel, we think what we think, because of the experiences we've had from the moment we're born to the day we die, and how those experiences impact our nervous system. I also believe, and that's known as experience-dependent plasticity, I also think understanding the fundamental mechanisms of the brain's plasticity is, hopefully obviously important, not only for understanding all the amazing adaptive functions of the brain, but also for understanding the pathology of the brain. So many of the mechanisms I'm going to talk about, if people want to, turn out to probably have relevance to certain neurodegenerative disorders. And I'll give you one example of how it may have relevance to complex neuropsychiatric disorders such as addiction. Okay, so when I think about experience-dependent plasticity, and those of you who came to my earlier talk know that I like to keep things as simple as possible. So when you think about the neural mechanisms that mediate experience-dependent plasticity, I would argue there are only three general mechanisms by which that can occur. That is, an experience can modify brain activity. One is via activity-dependent changes in synaptic strength, the strength of communication from one neuron to another. Another is structural changes in neural or synaptic connectivity. That is, activity-dependent changes in connectivity. That is the growth of new synapses, the pruning away of pre-existing synapses. I am making the assumption that people know what a synapse is. It's the physical connection between two nerve cells. It's the site at which two nerve cells in a nervous system communicate. And then finally, a topic that's received a little bit more attention, activity-dependent changes in the intrinsic excitability of neurons. And I would challenge any of you to give me additional mechanisms that, when all is said and done, don't impact on one of these three phenomena to mediate experience-dependent plasticity. For the first 30 years of my career, and even to this day, not quite to this day, I really focused on the cellular and molecular mechanisms responsible for activity-dependent changes in the strength of synaptic communication, which we term synaptic plasticity. Synaptic plasticity comes in two forms. You can increase the strength of synapses, the strength of that communication, and you can decrease it. We call these phenomena that we study in reduced preparations long-term potentiation of synaptic transmission, that is the strengthening of synaptic communication, or long-term depression, of synaptic transmission. And we use these acronyms of LTP and LTD. Mechanistically, most of our understanding of the cellular molecular mechanisms come from studying these phenomena in reduced preparations. A very common one is making a slice of a part of the rodent brain called the hippocampus. We started studying these synaptic plasticity phenomena in the hippocampus for two reasons. First is the knowledge that the hippocampus was an important storage site for certain forms of learning and memory. That comes from the famous case of patient HM, where an individual who lost their hippocampus, actually lost most of their temporal lobe, wasn't able to encode new memories. And then of equal importance, this preparation, which I don't even have a picture of, has technical advantages to studying synaptic weight changes. What you're seeing here is a very simple experiment. I don't think you did this experiment, but I'll give you credit, maybe you did, where we're stimulating a set of synapses, we're measuring the size of the synaptic response electrophysiologically. Then at this point in the experiment, we're giving a certain pattern of stimulation. And you can see the size of the response gets bigger, and it stays bigger in this case for an hour or so. That is synaptic plasticity. That is long-term potentiation of synaptic strength. This just shows if we give a different pattern of stimulation, we can actually make the size of the response smaller. That is, we can make this population of synapses weaker. And what this shows is that synaptic strength is under the bidirectional control of activity. That is, certain patterns of activity decrease synaptic strength, other patterns of activity increase it. And computationally, it is thought that this bidirectional, the ability of activity to bidirectionally control synaptic strength gives neural circuits and nervous systems enormous flexibility and power to encode new experiences and store those experiences. Now, these phenomenon of synaptic plasticity that are known by their rubric of by the acronym LTP and LTD, they're ubiquitous. It's very hard to find any part of the brain or any synapses in any part of the brain that don't express one or other form of LTP or LTD. I will say that all of the remainder of this talk, I am talking about plasticity at excitatory synapses in the brain that use the neurotransmitter glutamate, glutamatergic excitatory synapses. It turns out other types of synapses in the brain, like GABA synapses, which are inhibitory synapses, also show various forms of plasticity. But this is just a very partial list that wherever you look in the brain, activity can modify synaptic strength. This is just some articles I found. The truth is, this is a talk I really haven't given for about 10 years, so I'm just rebooting it. But it's very easy to find examples of phenomena that seem similar to LTP and LTD that we study in reduced preparations in human brains. Here you see titles of papers where people are looking at doing evoked potentials, sensory evoked potentials, then given certain patterns of stimulation and showing the size of those evoked potentials change for hours on end, even days or weeks. In fact, some of you may know that a very useful therapeutic modality that is getting a lot of more research done on it is transmagnetic stimulation. People are using that. This is stimulating certain parts of the brain using magnetic waves for therapeutic effect in depressed patients and all sorts of different patients. And some of the patterns of stimulation these investigators use are based on the patterns of stimulation people like me and others in the field discovered cause synaptic weight changes. So again, I'm trying to make the point that understanding these synaptic plasticity mechanisms may in fact have relevance to how the human brain works. Another reason for believing that, and again, this is actually an old slide, is that the genetics of most neuropsychiatric disorders, and again, I am a psychiatrist, so I mostly am referring to psychiatric disorders like schizophrenia, like obsessive compulsive disorder, autism spectrum disorder. If you look at the human genetic findings and the common variants that contribute risk to these various disorders, 60, 70, or 80% of the genes being implicated are, am I doing something wrong? Are actually synaptic proteins. So again, the idea, and some of these synaptic proteins play a role in these synaptic plasticity phenomena. So another reason why understanding these mechanisms may be of relevance to understanding higher brain function and pathology. And in fact, this is just an example of series of review articles. I don't know if you can see the titles, where this is one I wrote, Synaptic Plasticity and Addiction, which I'm going to talk about. I forgot to, I'm going to tell you that the forms of plasticity which we understand the best are triggered by a specific subtype of glutamate receptor known as NMDA receptors. And here's an article about the role of NMDA receptors in schizophrenia, et cetera, et cetera. So there's, reasonable data from human neuroscience and human subjects to suggest that these understanding the mechanisms of synaptic plasticity may be relevant. So when you're thinking about synaptic plasticity, you can divide the important questions one wants to address into two very broad categories. One, which is what I again studied for 30, 35 years, is what are the critical molecular mechanisms? How does it actually occur? And the other big question, which I'm going to touch on, well, the second half of the talk, is what are their in vivo functions? That is, who cares about these phenomenon I am defining as LTP and LTD if they never happen in the intact, awake, behaving brain in a manner that has functional or behavioral relevance. And I'm going to give you some examples that I think that hopefully support the claim that they do. So, for the understanding of molecular mechanisms, I'm now just going to take you through textbook knowledge. This textbook knowledge took, I have to think, 80s, 90s, took about 30 years to generate. And I'm going to summarize 30 years of research that I was involved in and probably 100 other labs around the world were involved in. And I'm going to focus on what in the field we call the classic forms of synaptic plasticity, and that is the forms of LTP and LTD that are triggered by a subtype of glutamate receptor known as the NMDA receptor. When you think about the mechanisms of plasticity, another way of simplifying that discussion is to talk about the induction mechanisms, that is, the mechanisms by which the synaptic weight changes are triggered, and the so-called expression mechanisms. That is, once these biochemical cascades are activated, what happens to actually change synaptic weights? And what this summarizes is that a whole body of work suggests that NMDA receptor dependent changes in intracellular calcium are a key component, I shouldn't say a key component, is the key trigger for synaptic weight changes for these forms of LTP and LTD. The other thing this slide points out to you is that what my work has been most relevant to and what I have been most interested in is so-called mediators of these phenomenon. And when I say mediators, I mean processes without which synaptic weight changes do not happen. They are mediators. They are required. It turns out there's many, many different ways of modulating these synaptic plasticity phenomena. And again, I sort of lifted this talk from 10, 15 years ago. In this field, historically, there was a lot of confusion about mechanisms and a lot of very intense arguments. And I think it was because the field didn't separate adequately the difference between processes and manipulations that modulated how you trigger and express LTP and LTD versus the actual core mechanisms. So when we think about the triggering, again, I'm going to summarize whatever it is, 20 years of work in a single slide. It's a little scary. This is a slide taken from a textbook written by my old friend Mark Baer, published 20 years ago. And again, this summarizes work I was very involved in back in the, back in the 80s and 90s. Several other people were involved. And again, the idea is, I'm sort of repeating myself, is that certain patterns of stimulation activate the NMDA receptor. The NMDA receptor is a filter for calcium. When it's activated and the cell is depolarized, calcium enters it. And that makes it very different than all other types of receptors in the synaptic membrane. And that when the pattern of activity is really strong, usually a very high frequency burst of stimulation, you get a very high level of calcium increase. And when the pattern of activity is at a low frequency or a certain type of activity, you get a very low level of calcium increase. And it's well established that to get LTP, you need this high level of calcium increase in the postsynaptic cell. In fact, in the postsynaptic dendritic spine, which is the morphological site where synapses are formed on most pyramidal neurons in the brain. So high calcium beyond some threshold triggers LTP. Some lower level of calcium, often a prolonged low level of calcium, seems to be the most efficient trigger for LTD. Then this high calcium activates a population of protein kinases. One in particular I'm going to tell you about in a second. And it's the activation of these kinases, in particular, I think it's in my next slide. No, it isn't. It's coming up. A kinase called calcium calmodulin-dependent protein kinase type 2. Notice the title. It's a calcium-dependent kinase. That seems to be a critical trigger for the increase in synaptic weight changes. And conversely, and I assume many of you know, kinases are enzymes that function by phosphorylating substrate proteins, and that phosphorylation of substrate proteins changes the function of those proteins. LTD, with this low level of calcium in contrast, seems to activate a cascade of protein phosphatases, including one that is also calcium calmodulin dependent called calcineurin. So the simple-minded hypothesis, and you can see I do say this was all work done in the 80s and 90s, is just what I said. LTP, high amounts of calcium triggered by NMDA receptor activation, activating calcium-dependent kinases. LTD, different patterns of activation at the synapse, causing a different type of calcium signal that preferentially activates phosphatases, and that these then phosphorylate certain critical substrate proteins in the synapse or dephosphorylate them, because that's what phosphatases do. And that, you know, I can talk for hours about the details of this and what's known and what's not known, but that gives you the high level. If you go into any textbook of neuroscience, I think you're going to see diagrams that look like that. So the second question is the expression. How does a synaptic weight change? And brief review of synaptic transmission. I did say this is not for a neuroscience audience. I'm hoping you have some knowledge.
Most people are actually, if not everybody, are neuroscientists.
Oh, they are. All right. Well, nevertheless, you're going to learn about synapses. So then you know, let me remind you, synapses work. Action potentials come down an axon, hit the presynaptic Buchanan terminal, neurotransmitter is released, diffuses across the cleft, binds to receptors, in this case glutamate receptors, to trigger a current that generates the excitatory postsynaptic response. So given that, if you were my students, I'd ask you, how could you increase synaptic strength or decrease it? There's really only two mechanisms. One is by changing the amount of neurotransmitter release. Less neurotransmitter release, you have a weaker synapse. More transmitter release, a stronger synapse. The other is by modifying the postsynaptic receptors that are responding to a fixed amount of neurotransmitter release. Now, if you think about that, and you think about the first mechanism, which is a change in neurotransmitter release, remember I just showed you that the triggering of these forms of plasticity are postsynaptic. They're happening in the postsynaptic side of the synapse. If there is a change in transporter release, that requires some form of retrograde messenger, some signal that goes from the postsynapse back to the pre. And suffice it to say, there was a 20-year fight. It was really an argument and a fight between some very prominent neuroscientists. I don't know if I was prominent, but I was one of the people battling the fight. The pages of Nature and Science were full of papers discussing this very simple issue. That is the expression mechanisms of LTP and LTD. And I'm happy to tell you about they used to be called the LTP wars. And suffice it to say, I think most people have come around to accepting the following mechanism for LTP. And let me see if this is going to work. Oh, it worked so. Whoops. Hold on. How do I get a movie to work with a laser? I worked so hard on this movie. Hold on.
Yeah, I think you need the...
What do I do?
You need the regular pointer back and then put the laser pointer back.
Wait, get rid of the...
Yeah.
All right. So what I am showing here, thank you, whoever just told me that, is the simple-minded idea, which I'm proud to say I was a major contributor to this idea, was that the way LTP works is there's a population of AMPA receptors that are inside the cell or adjacent to the synapse. And following the activation, the induction of LTP, these receptors get inserted into the membrane and the synapse gets stronger because there's now more receptors in the membrane. And conversely, LTD involves the removal of receptors so activation of this phosphatase cascade and a whole bunch of other biochemical signaling mechanisms results in pulling away a population of receptors known as AMPA receptors due to a process known as endocytosis, and the synapse gets weaker. And in fact, Ami Citri, when he was a postdoc in my lab, did some interesting work on some of the molecular mechanisms that kept these receptors inside the cell during LTD after they were endocytosed. And again, I think this is now textbook knowledge. Most textbooks will report this. So if you put this all together, you can get a very simple model of this specific form of LTP and LTD. That is LTP, again, a big rise in calcium, activating a kinase, calcium calmodulin kinase 2, causing the exocytosis or delivery of AMPA receptors to the synapse and making the synapse stronger. LTD involving a lower increase in calcium, activating a phosphatase cascade that includes a calcium calmodulin-dependent phosphatase known as calcineurin, that leading to the removal or endocytosis of AMPA receptors. And this is known as AMPA receptor trafficking. These hypotheses were put forward in the mid-90s. It led to 20 years of work and working on the detailed molecular mechanisms, and people are still working on these kinds of mechanisms. The one other thing I'm going to say about synaptic plasticity is that LTP and LTD also it's clear now, involves structural changes to the synapse. As most of you know, glutamatergic excitatory synapses, at least when they're formed on pyramidal neurons, not necessarily GABAergic interneurons, but projection neurons in the cortex and the hippocampus, they form on these protuberances known as dendritic spines that come in these interesting and perhaps beautiful shapes. And it's very well established, these are not the best pictures, that if you image spines in living brain tissue, and this has actually been done in vivo, that LTD involves the loss of spines. If you look at this spine, it's undergoing an LTD protocol, and it sort of has shrunk away. And let me see if I can give you, if you look at this spine, it's not as clear an example. Pardon me?
You don't see your point.
Oh, I'm sorry. Thank you. I see it on my screen. If you look at this spine, you give it an LTD protocol, and it slowly kind of disappears. It's gone from a mushroom thing to this little thing. This isn't the best example, but this spine is undergoing LTP, and you can see it sort of gets bigger and thicker. And so there are structural changes that accompany this AMPA receptor trafficking changes. And furthermore, actually, there was just a paper in Nature like a few weeks ago showing that even in the adult rat brain, you can actually generate new synapses via kind of LTP-inducing protocols. So the thinking is that these synaptic plasticity phenomenon not only cause functional changes in synaptic strength, but also are accompanied by structural connectivity changes. And then finally, just to end this first part of the talk, I've only been talking about the major forms of synaptic plasticity. That is NMDA receptor-dependent LTP and NMDA receptor-dependent LTD, as most extensively studied in the hippocampus, in particular at excitatory synapses on CA1 pyramidal cells. Over the years, it's become very apparent that there are other forms of synaptic plasticity, and that have funny names based on where they're found, like cerebellar LTD or mossy fiber LTP, or the receptor that triggers them. So there's something called metabotropic glutamate receptor LTD, because rather than being triggered by NMDA receptors, it's triggered by a different subtype of glutamate receptor. There's a form of plasticity that involves the release of endocannabinoids, et cetera, et cetera. So enough said. So that's a whirlwind tour. If you're a neuroscientist, I didn't tell you anything you didn't know, hopefully. But hopefully it was a decent review. So the next question is, what are the in vivo functions? Like, how do you actually explore whether LTP and LTD actually occur in the awake living brain and play a role? And here's a little bit of history. These phenomenon were first primarily described, well, LTP in particular, but even LTD, in the hippocampus. And as I mentioned, it was very exciting in the 70s when LTP was first described by Terry Lomo and Tim Bliss, because the hippocampus was known to be important for learning and memory. And people as far back as Donald Hebb had proposed theoretically that synaptic weight changes are an attractive mechanism for encoding new memories. But for the next 20 or 30 years, LTP was first described in 73. Really, for the next 20 years, even 30 years, people kept trying to do experiments that would show LTP occurred in the hippocampus and was important for learning and memory. And it was really hard to do. There were hints. There was a paper here, a paper there, but in fact, there was no compelling data. It's gotten better now for a variety of reasons I'm willing to talk about using the modern tools of neuroscience. But in the when was this? In the mid to late 90s, I was working on LTP and LTD, and I was worried about that issue. I was worried that maybe I'm studying a synaptic artifact, an artifact that was still interesting to study, that could still tell me something about how synapses work, but these artificial phenomenon in a hippocampal slice of LTP and LTD, maybe they never happen in the brain. I then decided that the hippocampus might not be the best place to look, and the reason people were having so much problems finding examples of synaptic weight changes in the hippocampus is because the hippocampus uses a very sparse coding mechanism. So if a memory is stored in the hippocampus, it probably involves synaptic weight changes broadly distributed among hundreds of different neurons, and each neuron has 15,000 synapses, and maybe only a few synapses on an individual neuron are being modified, and it's just going to be impossible to find the synaptic weight changes that occur during hippocampal-dependent forms of learning and memory. So I decided to shift up, and again, I showed this slide in my earlier talk. I was influenced by this very simple-minded thinking that that behavior could be explained by positive and negative reinforcement, or positive reinforcement versus aversive learning. And I started thinking, where might I be able to, what sets of synapses, what parts of the brain might I be able to do an experimental manipulation that would cause a behavioral change that might involve a synaptic weight change that I would be able to detect. And I started thinking about stimuli that were rewarding, thinking maybe I could give animals a rewarding experience and look for synaptic weight changes in mesolimbic dopamine circuitry. Suffice it to say, I started reading the literature on drug addiction. This is in the mid-90s, late 90s. And it turns out that for a variety of reasons, neuroscientists studying the neuroscience of addiction had begun to propose the idea that drugs of abuse cause some of their behavioral changes because they elicited synaptic plasticity in key nodes of the mesolimbic reward circuitry. And the basic idea goes something like this. That the bottom line is that the persistence of addiction reflects in part drug-associated memories in the form of altered circuitry due in part to drug-induced synaptic plasticity. Let me just see. And the strongest evidence in support of that idea, again, I've showed this in my 11 a.m. talk, and again, I assume all of you know this is the mesolimbic reward circuitry in primitive form. VTA dopamine neurons are found here. They project to the nucleus accumbens. And so I was reading the literature, and there was a series of papers reporting that if you infused antagonists to the NMDA receptor into the VTA, okay, so you're blocking NMDA receptors in the VTA, the home of dopamine neurons, you could block some of the behavioral effects of drugs of abuse. And remember I said I was studying NMDA receptor triggered forms of plasticity, LTP and LTD. So to somebody like me, that screamed perhaps drugs are acting in the VTA to cause synaptic weight changes. And again, some of the people in the field had proposed that, but because they were not synaptic physiologists, they didn't know how to study it. They were measuring extracellular glutamate, they were making claims. And so I came into the field and just did, at the time, work. They were new experiments, but they were, how to say this politely, they were obvious experiments to do. The field just didn't know how to do them. And the experiment was very simple. And I did this in collaboration with Antonello Bonci, who was a postdoc in my lab, and then he started his own lab, and we did it in collaboration. It was very simple. We just took mice, we gave them a drug, either saline or cocaine, we waited a day, we cut a slice that contained the ventral tegmental area and dopamine neurons. And then we made recordings from those neurons, and we measured synaptic potentials. And the big insight we had, which I will take credit for, is how do you compare synaptic weights at excitatory synapses between different animals? One animal got saline, one animal got, in this case, cocaine. And I came up with this idea of comparing the ratio of synaptic responses mediated by a subtype of glutamate receptor called an AMPA receptors as normalized to the amount of current being generated by NMDA receptors. And this became known as the AMPA-NMDA ratio. And the reason I came up with that idea is that obviously it's a ratio. So if both components of these currents that you're measuring increase or decrease together, the ratio is not going to change. But from hippocampal work, we knew that classic LTP only involved an increase in AMPA receptors, but not NMDA receptors, as those films I showed you. So the idea was if cocaine was actually causing an LTP-like event at excitatory synapses on dopamine neurons, we should see an increase in this AMPA-NMDA ratio, and obviously we did. So that actually said that an in vivo experience of cocaine was likely triggering an LTP-like phenomenon in vivo in an awake behaving animal at excitatory synapses on dopamine neurons. We then went on to show that this same synaptic weight change, this increase in the AMPA-NMDA ratio, happened in response to many different classes of drugs of abuse, amphetamine, morphine, nicotine, alcohol, but it did not occur in response to non-addictive drugs like Prozac, fluoxetine, or carbamazepine, which is an anticonvulsant. So it wasn't as if any drug would cause this change. It was only drugs that had abuse liability. And that was among the first evidence that LTP, LTD, well, in this case, LTP-like phenomenon could actually occur in the brain. This is just a cute experiment. Antonello Bonci went on to do this experiment where the strategy is the same. He teaches animals to self-administer now. So, they press a bar, they nose poke to get cocaine, to get food, to get sugar. And if he does this and he looks at synaptic weight changes on dopamine neurons just a day later, they all cause a synaptic weight change. But only the cocaine causes a synaptic weight change that lasts 3 weeks or 3 months, causes a very long-lasting change. And I would like to believe this kind of work, which is now done, began over 20 years ago, has had a major impact on the addiction field, because then everybody started doing kinds of similar experiments, and then they started incorporating optogenetics in ways that I'm going to talk about in the next 15 minutes of my talk. So that's all well and good. So where did we go with this? How did this help us build further understanding of synaptic plasticity mechanisms and the circuits in which synaptic plasticity occurs? Well, as all of you know, dopamine neurons come in different flavors. The most simple distinction is there are dopamine neurons in the pars compacta of the substantia nigra that send projections to the striatum. There are dopamine neurons in the VTA. Some of them send projections to the nucleus accumbens, some to the prefrontal cortex, some to the basolateral amygdala. So now we're up to like 2009 or 2010. And a postdoc named Stéphane Lamel joined my lab. He's now a tenured professor at UC Berkeley. And Stéphane, for his PhD, had done work showing that dopamine neurons in the VTA are heterogeneous. And as I said, they don't all project to the same targets. Some project to one target region, some project to others. They have different biophysical properties. So that made me wonder whether the synaptic weight changes in response to a rewarding experience or an aversive experience might cause changes, different changes in these different subpopulations of dopamine neurons. And so Stefan came to my lab, and he used the technique he had used for his PhD, where in different populations of mice, he injected retrograde fluorescent beads in the prefrontal cortex, in the certain regions of the accumbens, in the dorsal striatum. He waited a while. He then gave the animal an experience, either cocaine or an aversive experience, which I actually can't even remember what it was. I think it was an injection of formalin into the hind paw, painful experience. He then cut slices so he could record from dopamine neurons, knowing which dopamine neuron he was recording from, knowing whether that dopamine neuron projected to the prefrontal cortex versus the nucleus accumbens versus the dorsal striatum. Okay? So this is a complicated slide. This summarizes a neuron paper Stefan published with me now, whatever it is, 12 years ago. And the key results are here. So here are the different populations of dopamine neurons he recorded from. The ones in the pars compacta that project to the striatum, the one in the lateral VTA that project to the nucleus accumbens lateral shell. There's ones in green here that project to the medial shell. There's ones in red that preferentially project to the medial prefrontal cortex. And here are the key results. That when he gave the animals cocaine, only the dopamine cells that projected to the lateral or medial shell showed an increase in synaptic weights as measured by the AMPA-NMDA ratio. Surprisingly, the dopamine neurons that projected to the PFC did not show this synaptic weight change in response to cocaine. Instead, they actually showed an increase in the synaptic weight, in plasticity, in response to a highly aversive experience. So what this says is that synaptic weight changes in this heterogeneous population of dopamine neurons has some specificity. It also suggests that all dopamine neurons are not created equal, that they may subserve different functions. And in fact, if you start going into the systems neuroscience literature of people who record single unit activity from dopamine neurons in awake behaving monkeys or rats, of course there's the classic dopamine neuron response first described by Wolfram Schultz, which encodes reward prediction errors. I am not doubting that. And if you don't know what I'm talking about, it's not that important. It's just dopamine neurons that respond to novel rewards or cues that have been associated with rewards. But even back in the early 2000s, it wasn't that hard to find examples of dopamine neuron recordings where the dopamine neurons responded to aversive stimuli, not rewarding stimuli, or they responded to both aversive and rewarding stimuli, indicating that they were neurons that responded to the salience of the stimuli, not necessarily the rewarding. So we looked at this literature. Stefan and I were intrigued by this set of findings, that synaptic plasticity seemed to be, the type of plasticity we saw seemed to be cell type specific and based on the type of experience the animal had. So that raised the question of, if these dopamine neurons are projecting to different regions, maybe the inputs onto the dopamine neurons might be somewhat different, because it didn't make sense to us that if the dopamine neurons were subserving different functions, they would all have the same set of inputs. And the truth is, I'm not going to present this, I've done a lot of work with Li-Chun Luo using modern tracing methods to really expand upon that idea. What I'm going to present to you now in the last 10 minutes of my talk is the initial foray into this question of the input specificity of inputs into different populations of dopamine neurons. And this work was done by Stefan Lell in collaboration with a postdoc who's now a professor at UCSD, Byung-Kook Lim. And what BK did is he read the literature, and BK is very creative. He created a modified rabies virus that allowed, and rabies virus is basically a retrograde label. It's taken up by terminals in the site in which you inject it. It's transported back to the soma. If the rabies virus is expressing a transgene like GFP, it turns the cell green, but it can't replicate anymore, so it doesn't keep going. So it only will label the cells that are sending direct presumptive monosynaptic connections onto the cells in the brain region where you injected the rabies virus. So this was just a way of sort of looking at all the different inputs, sending projections into the VTA. And here's what the data actually looks like. And then you can look in different brain regions and see green cells. So we now knew there were all these different brain areas that sent inputs to the VTA. How were we going to activate those and manipulate them and study them? And again, I thought this wasn't going to be a neuroscience audience. That's what I was told. So I have slides about the awesome power of optogenetics. But does everybody know what optogenetics is? All right, so I don't have to go through. I just saved 2 minutes. That we used optogenetics. I was fortunate. I'm bragging here. Carl Deiseroth was my postdoc. He started doing optogenetics like the week after he left my lab. Like any good postdoc, he never told me what he was doing. This is all true. Carl is a very close friend, so I know the whole story. So he created the field of optogenetics, and that then allowed us to kind of do the obvious experiment. We now expressed channelrhodopsin in our rabies virus. We could inject it into the VTA. All the cells that send inputs into the VTA now have channelrhodopsin. So we could then pick some inputs we wanted to activate and ask what were the behavioral consequence of activating specific inputs using optogenetics, specific inputs into the VTA. So we chose 2 sets of inputs for a variety of reasons. I'm just using these as examples. Again, this paper was published over 10 years ago. For a variety of reasons, both based on the literature and some other factors, we decided to compare the effects of activating inputs from the lateral dorsal tegmentum, the LTD, versus now what has become a famous structure, the lateral habenula. And the experiment we did was a simple conditioned place preference experiment. We took the mice, we put it in a 3-chamber, house. We conditioned it with optogenetic stimulation in one chamber but not the other for a day or 2. And then we brought the mouse back and asked, which chamber do you want to hang out with the assumption that if the activation of the set of inputs was reinforcing, we could get a conditioned place preference. The animal would prefer to hang out in that chamber. If the input activation was aversive, we would get conditioned place avoidance. And what we found was that if we activated the inputs from the lateral dorsal tegmentum, we got a very reasonably robust conditioned place preference, saying that the activation of those inputs was reinforcing. But at the time, this was surprising. Now it's common knowledge. But at the time, this was the first evidence that if we activated the habenula inputs into the VTA, it was fairly aversive. The animals showed conditioned place aversion. So then, in that initial set of experiments, this was all being done in vivo, actually. So in these experiments, we were activating the cell bodies of the lateral dorsal tegmental neurons, or the cell bodies of the habenula. That doesn't tell us that it's the actual inputs in the VTA that are mediating these effects. So we repeated these experiments, but now we're shining the light in the VTA. So we're activating the inputs from the lateral dorsal tegmentum or the inputs from the lateral habenula, and now you see the results were even perhaps more robust. The activation of the inputs from the LDT were reinforcing, gave us very robust condition place preference, and the activation of the habenula inputs was highly aversive. We got a condition place aversion. So accepting these behavioral results, by definition, that means these inputs must have different connectivity within the VTA. If they're both glutamatergic inputs. So if they were synapsing and influencing the exact same population of neurons, we should have gotten the same behavioral results. In fact, we didn't. So we then did some slice physiology. The details aren't important, but again, we took Stefan's approach where we retrogradely labeled populations of dopamine neurons using retrobeads. And here's just an example of what it looks like. It's a pretty labor-intensive effort. We expressed channel rhodopsin, in this case, in LDT neurons. And then we did slice physiology. So what Stefan was able to do is optogenetically activate only the LDT inputs while making recordings from dopamine neurons, knowing which target they projected to. And the key result is really shown in these two green graphs. When he activated the LTD inputs, he always got a synaptic response in the dopamine neurons that projected to the medial shell of the nucleus accumbens. The size of the responses was pretty good, and the percentage or degree of connectivity was very good. And in fact, if he recorded from dopamine neurons that projected to any other brain region, most importantly, the prefrontal cortex, the inputs from the LDT barely made any synapses on those dopamine neurons. So there was a preferential connectivity from LDT inputs onto dopamine neurons that projected to the shell of the accumbens. He then did the same type of experiment in the where he retrogradely labeled dopamine neurons, activated, in this case, lateral habenular inputs, and in addition to recording from dopamine neurons, he made some recordings from an adjacent area called the RMTG, because there was already some evidence that the habenula may be sending projections to the RMTG, which is a set of GABAergic neurons that then inhibit dopamine neurons. So you can see where this is going. And most importantly, what he found is, if you look at these two graphs, is when he activated habenula inputs, and I'm almost done, he saw no connectivity to the dopamine neurons that the LTD inputs were synapsing on, but he found very strong connectivity onto the dopamine neurons that project to the prefrontal cortex and onto these GABAergic neurons in the RMTG that we subsequently show make inhibitory synapses on a different population of dopamine neurons. Now, the reason this is interesting is, remember what I told you, is that the dopamine neurons that project to the prefrontal cortex, their glutamate synapses did not show a synaptic change in response to a rewarding experience, cocaine, but did show a synaptic weight change in response to an aversive experience. So I'm going to skip this. This just shows you that the behavioral effects of activating LTD inputs versus habenula inputs do involve changes in dopamine levels in target structures. Here's the final key experiment. So the hypothesis was that lateral habenula input activation in the VTA was causing an aversive experience, condition place aversion, for two reasons. It was preferentially activating a subpopulation of dopamine neurons that project to the prefrontal cortex. I didn't show you data to support this next statement, but we also knew simultaneously the lateral habenula inputs were driving GABAergic neurons in the RMTG that were then inhibiting the dopamine neurons that are normally reinforcing, that normally send projections to the accumbens. So the prediction of this would be that if we activated the dopamine inputs in the prefrontal cortex, that should be aversive. And that's what's shown here. If we actually did that experiment, this was done a few years later, in collaboration with Lisa Gorniden, who is a postdoc in Karl Deiseroth's lab. If you activate dopamine inputs in the PFC, the animals express conditioned place aversion. And finally, the reason this might be a little relevant is based on some of this work, not only our work, but the literature, a colleague who was then at Stanford, who was an fMRI imager, who did studies of resting state or task-evoked fMRI measurements, looking for networks that were activated by certain experiences. In part, based on Stefan's work, he looked at the fMRI responses focusing on the habenula and VTA. And what this shows you, and it's controversial because some people think you can't image the VTA using fMRI in human beings. Sam McClure claims he did. The bottom line is when he gave the subjects an aversive stimuli, he saw task-evoked increases in the bold signal simultaneously in the habenula and the VTA. So at a high level, this is at least consistent with the kinds of circuit hypotheses we were generating in the mouse. So my last slide is very confusing. It's not critical that you understand this. The idea just being how one could go from studying synaptic plasticity mechanisms to looking for in vivo experiences that elicited synaptic plasticity, then look at the cell type specificity of those synaptic weight changes, and based on that, start using the modern tools of circuit neuroscience to begin to try to elucidate in a more sophisticated way circuit motifs or circuit connectivity that mediate reward and aversion. And again, the details are not important. This is just the circuit map we came up with 10 years ago. And what I will finally say is I began this by talking about what were the circuits in the brain that mediate reward and aversion. Of course, it is going to be complicated. And I always like to show, I'm just focusing on one component of the brain-wide mechanisms that probably encode and implement our responses to highly rewarding and highly aversive stimuli. I'll skip that. I'm out of time. Anyhow, thank you for your attention, and I'm happy to answer any questions.
Yeah, so a couple of points about the beginning of your talk. Mm-hmm. So first of all, I noticed that when you were showing how calcium comes in, did it only show the NMDA receptors and not voltage-gated calcium channels? Is that intentional?
It is, but it turns out, I did a lot of work on that. I mean, you can drive LTP and LTD with activation of, at least in hippocampal pyramidal cells, of L-type voltage-dependent calcium channels. The assumption, but you have to drive them really hard. You have to do whether LTP stimulation is physiological, who the hell knows, but you have to drive the spiking, you have to activate voltage-dependent calcium channels, usually L-type, very strongly. And the thinking, without much evidence, although it makes sense, is that all the biochemical signaling mechanisms for traditional LTP-LTD is in the dendritic spine. The L-type calcium channels probably aren't in the spine. They might be in the neck, they might be in the shaft. But if you drive them hard enough, you can probably get enough calcium entry that it dribbles into the spine and starts activating the same mechanisms. The reason we say that is I have a paper or two, a neuron paper actually with Jennifer Cummings, ancient history from the 90s, showing occlusion, if you know what occlusion means, meaning if we induce LTP with voltage-dependent calcium channels, it was actually LTD, we no longer could get regular LTD via NMDA receptor activation, suggesting they were activating the same mechanisms. But I think for the most part, my hunch is in the intact, awake, behaving nervous system, NMDA receptors are the critical trigger. And that comes from all the work using pharmacological antagonist of NMDA, using conditional knockouts of NMDA receptors. You lose plasticity pretty much, these forms of plasticity.
Right, but the idea might be that you have both of them at the same time, right? So for example, in order to do STDP, so you might have to have like backpropagating action potential to open up multiplication calcium and energy.
Yep, and that is certainly a possibility. I'm not sure how strong the evidence is that STDP requires L-type calcium channels. I always have had a really hard time getting STDP to work, but I know the computational people love it because it's such a beautiful model for triggering plasticity. So yeah, so I wouldn't argue with you. I think it is a source of calcium that can trigger synaptic weight changes. I have no doubt about that.
Yeah, my second point is maybe a more high-level question, but like in principle, what you need to do for plasticity is fairly simple. You either increase or decrease the synaptic weight, right? So you either insert or remove AMPA receptors. So why are the molecular mechanisms and all these like structural proteins, why is it so complicated for such like simple process?
First, is it that complicated? I don't know. Remember my whole talk about mediators versus modulators.
Yeah.
So there may be lots of ways of modifying your ability to get LTP or LTD that are not part of the core mechanism. That's one hand-waving argument. The other is they are so important, you want enormous redundancy so that when you have genetic mutations in one set of proteins or complex signaling cascade that under this circumstance is required for plasticity, you have another mechanism sitting right there to make up for it. And in fact, this is not about plasticity, but when I used to collaborate with Tom Sudhoff, I'd done a lot. I mean, Tom and I, it's mostly Tom making the knockouts, and then I get to study them. It is really hard. It's actually he has never found a genetic manipulation that prevents synapses from being formed and functioning. I mean, we actually unpublished. When I say we, he made a conditional knockout of 7 synaptic cell adhesion proteins, all the neuroligands and all the LRTMs, which is a certain form of synaptic cell adhesion protein that he and I had studied, and it's involved in maintaining AMPA receptors. Think about that. We got rid of 7 synaptic proteins with the hope that the synapse would just barely be functioning, and it was pretty normal. So I hope I'm answering your question. But it's hand waving. I don't know.
Yeah.
I bored you to death. That's okay. It's time for a beer. Any other questions? About anything? All right. Well, thank you for your attention.